Hello, everyone. My name is Amanda Olbrick Marabesi, and I'm a PhD candidate in horticulture at the University of Georgia. I really appreciate the invitation to give a talk for the CRC group. And today I'm gonna talk a little bit about my PhD research, which the overall theme is the influence of cadmium exposure on industrial hemp grown for cannabinoid production. And here on the left are my committee members, my professors who have supported me throughout this PhD journey. So a little bit about hemp, which of course you guys know. So hemp is, used for industrial or medicinal use. And for the medicinal use, it's usually sold as biomass and smokable flour. So the biomass for the oil extraction, right? And cannabinoids like CBD and THC are the compounds of interest in hemp. So heavy metal concentrations in hemp biomass or in the smokable flour is an area of concern as the products may be consumed as dietary supplements. So a little bit about cadmium now. Cadmium, it's a heavy metal, and humans can be exposed to heavy metals via water, food, cosmetics, medicinal products, and smoking. And cadmium has been associated with smoking tobacco because tobacco is a cadmium hyperaccumulator, so it can uptake cadmium from contaminated soil and accumulate it in its leaves. So then when people smoke that contaminated tobacco, they're also inhaling all the cadmium that is present in the leaves. There are harmful effects of cadmium inhalation, which include renal tubular dysfunction, osteomalacia, which is a disease of the bones, lung disease, and it's also classified as a group 1 carcinogen. However, consumers are less likely to perceive the harms of cannabis smoke to the same extent as the harms caused by smoking tobacco. And heavy metal testing in hemp is currently not federally enforced, and there are inconsistencies with heavy metal limits in different states where hemp is grown and processed. So pretty much each state now has its own regulations regarding heavy metal testing. Some states test, a number of different heavy metals, while other states test only a few, and while other states don't test any. So here's an example from the California Bureau of Marijuana Control. So they have 3 categories of medical cannabis goods, and they test for 4 heavy metals, which are called the Big Four: cadmium, lead, arsenic, and mercury. So here, for example, for inhaled medical cannabis goods, which would be the hemp smokable flour, the level for cadmium, the action level for cadmium is 0.2 parts per million. About heavy metals in ag soils. So cadmium can be introduced to agricultural soils in contaminated manure and biosolids, phosphate fertilizers, mine waste, atmospheric deposition, and industrial waste. And although the potential harms, to ecosystems, human, animal health caused by heavy metals have been researched, there are still inconsistencies regarding what are the metal thresholds for ag soils. And the U.S. EPA has set no thresholds for total or bioavailable concentrations of heavy metals in agricultural soils. They do have thresholds for residential soils, but nothing for ag soils. So there's clearly a need for research. Hemp has the agronomic traits that could facilitate accumulation of heavy metals. They have an extensive, deep root system, efficient translocation of contaminants. So the same way they uptake a lot of water and nutrients from the soil, they could potentially uptake contaminants. They have a rapid life cycle and high biomass production, which could also contribute to that. However, there's a lack of research on metal accumulation by the hemp varieties that are destined for human consumption. So there's a lot of research published on hemp fiber varieties. How can they be used for phytoremediation of soils? But there's not much on the varieties grown for cannabinoid production. So this is the first chapter of my dissertation, and it has been published in Frontiers in Plant Science journal. And this is what I'm going to focus on today. Cadmium exposure is associated with increased transcript abundance of multiple heavy metal associated transporter genes in roots of hemp. So the objectives of this study were to evaluate the processes of uptake, translocation, and accumulation of cadmium by the industrial hemp variety Purple Tiger, which is a variety from the hemp mine. Dr. Justice kindly gave us cuttings of the Purple Tiger, and I propagated them for my experiment. So thank you, Dr. Justice, again. And we did this experiment in 2 seasons, which I call them experiment 1 and 2. So we grew the plants in a deep water culture system, which I'm going to show you next, containing a nutrient solution and a non-concentration of cadmium. So we had 4 treatments. The control treatment which had no cadmium. Then we had 2.5, 10, and 25 milligrams per liter of cadmium. So here's a little bit of the propagation. Here are the mother plants that we had. So all Purple Tiger plants, we had them in a greenhouse. And I just used, took the cuttings, used Clonex, and then stuck the cuttings in those, they're called Oasis foam cubes for rooting. And I think it took about 3 weeks for them to have a good root system for me to start the experiment. So here's pictures of the deep water culture system. Is a hydroponic system. So we placed the cuttings in 4 cuttings per tub. So each tub was 1 rep. We made holes with a drill and put those plastic baskets, which we could put the cuttings, the rooted cuttings. Here on the right, you can see that each tub is connected to an air pump. So the tubs are not connected to each other, but they are all connected to an air pump and then an air stone inside each tub to provide oxygen to the water, to the solution. We also have, you can see on the corner right, no, on the corner left, I'm sorry, sensors to monitor temperature, humidity, and light intensity. And what else? So yes, each tub was an individual rep, and we had a complete randomized block design. Here's a quick video showing how I managed the system. So I had to take care of each tub individually. So here in this video, I'm just adjusting the pH of each tub. So nothing in the system was automated. So for everything, from adding nutrients, adding cadmium, or adjusting the pH, monitoring the pH, or checking the EC of the solutions, I had to do everything individualized for each tub. Okay, here's a bigger picture of the system. We also had this supplemental light on top of this experiment. This was just to provide enough light for the plants to keep vegetative until we wanted them to flower. So it was not intended for growth or anything. It was just for to manage the photoperiod of the plants or manipulate the photoperiod. So the plants, after placing the rooted cuttings in here, they stayed in nutrient solution only for 4 weeks for acclimation before we introduced the cadmium to the tubs. We just wanted the plants to be a little bit bigger, have a bigger root system before we introduced the cadmium. So a little bit of materials and methods. A new nutrient solution was prepared at every 14 days, aiming 150 ppm of nitrogen. And we used just a commercial fertilizer, NPK, for that, dissolved the fertilizer in the solution. And then every 14 days, I had to dump everything out of the tub and start a new one. And the solutions were analyzed at the beginning and end of every cycle. And here a cycle would be the 14 days. EC and pH of the nutrient solutions were monitored weekly, so we targeted 5.5 to 6. Although I read many articles saying that cannabis can, be grown up to 5.6. I mean 6.5 pH, but we try to keep in that 5.5 to 6 range. We assessed photosynthetic yield and chlorophyll content index after 14, 45, and 68 days after exposure to treatments, which we're going to call DAT, days after treatment. I collected root, stem, leaf, and flower material at harvest for dry biomass quantification, cannabinoid analysis, and metal content analysis, and also collected root, leaf, and flower material at 3 time points throughout the experiment for my gene expression analysis. All my stats were conducted using JUMP and one-way ANOVA with Tuckey's HSD test for mean separation when appropriate, and when necessary, I log transformed the data. So here are some cool pictures. This is 2 days after introducing cadmium to the solutions. So you can see on the left, the 10 milligrams per liter of cadmium, and on the right, the 25 milligrams per liter of cadmium. The plants started showing this kind of like clawing of the leaves symptoms, and you can see how it's more pronounced on the higher cadmium treatment. And this was only 2 days after introducing the cadmium, so you can see how plants respond very quickly to the treatment. You can also see some chlorosis on the new leaves. And compared to the control on the left, which showed no symptoms at all, you can see the 10 milligrams per liter on the right. And also comparing the 2.5 milligrams per liter, which didn't show any symptoms at all, to the 25 milligrams per liter cadmium. So control and 2.5 milligrams, there was no symptoms, but 10 and 25 milligrams per liter, they show this clawing, curling of the leaves. This is 11 days after treatment, and we can see in the 25 milligrams per liter cadmium, the new leaves are showing some marginal necrosis and just like a generalized chlorosis on the older leaves. Overall, you can see that this plant is struggling. Here's comparing the roots of the control on the left versus the 25 milligrams per liter cadmium on the right, 11 days after treatment. So you can see how the 25 milligrams per liter of cadmium is causing this kind of like browning of the roots, and also the roots, they have a lot less volume compared to the control treatment, and this is only 11 days after. Here you can see also the air stone. It's in the tub, aerating the solution. And also you can see in the tubs, on the side of the tubs, there's an electrical tape on the right side of each tub. And this is where I kept the tubs with constant volume. So every 2 to 3 days, I would fill up the tubs with water to keep a constant volume in each tub. So here's 30 days after treatment, a picture of all treatments side by side. So from left to right, we have 25, 10, 0, which is the control, and 2.5 milligrams per liter of cadmium. I think that the three treatments, control, 2.5, and 10, are very hard to distinguish visually. Maybe the 10 has a little less branching, a little less biomass overall, but the control and the 2.5, you can't really tell. But you can see for sure that the highest cadmium treatment is struggling. The plants are very small compared to the other ones. They're yellow, they're necrotic, so they were struggling. That's why 45 days after treatment, we had to harvest the highest cadmium treatment. The plants were looking like this, they were barely alive. And since I was going to do gene expression analysis, I cannot use that tissue for that. So we decided to go ahead and harvest them. They were not able to form any flowers, any inflorescence, and their root system was very slimy. It was pretty much dead here for this specific plant. And it looked a little bit like Pythium, like very slimy, and it would break very easily. And then finally, at 68 days after treatment, we harvested the 3 remaining treatments. That was the 8th week of flowering, and we decided to harvest on that week because Dr. Justice recommended this between the 8th and 9th week of flowering for this specific purple tiger variety. Okay, so now a little bit of the results on plant growth and yield. So for plant height, we didn't see any differences among the 0, 2.5, and 10 milligrams of cadmium per liter, but then we saw a significant decrease in plant height for the 25 milligrams per liter of cadmium, and you could see that in the pictures too. Let me see if I can get the pointer. Okay, I don't know how to do that, but anyways, technology, right? So for the dry weight of plants, we saw some differences, but mainly in the 25 milligrams per liter of cadmium. So here we have stems, roots, and leaves plus flowers. We quantified leaves plus flowers because that's following industry standards. That would be the equivalent to biomass. So here we didn't see any differences for the biomass in the 3 lower cadmium treatments or roots or stems even. So we really only see a decrease in dry weight for the 25 milligrams per liter of cadmium. Some cadmium concentration results for plant tissues. So we quantified cadmium in flowers, roots, leaves, and stems for all treatments. And in this table, we have data for experiment 1 and experiment 2. So what I want y'all to focus on is on the amount of cadmium that we found in the flowers. So you can see that cadmium concentration increased with cadmium in solution. But in both experiment 1 and experiment 2, there was a lot of cadmium in the flowers. Even for the 2.5 milligrams per liter of cadmium, which the plant showed no symptom, no reduction in plant height, no reduction in dry weight, we saw like 8.7 to 7.6 milligrams per kilogram, which would be equivalent to ppm. And if you remember in the beginning, I said that the action level in California for inhaled cannabis goods, which would be the hemp flower, was 0.2 ppm. So this is like 40 times over the action level. Nevertheless, most cadmium stayed in the roots in both experiments. And also, it's important to emphasize that even the 2.5 milligrams per liter of cadmium, that's a lot higher than what would be encountered in agricultural soils, especially because everything that is in solution is available to the plants. While maybe you have 2.5 milligrams per kilogram of soil total, but it doesn't mean that all of that would be bioavailable to the plants. So, the levels that we used in this experiment are a lot higher. Nevertheless, we see that the plants are able to, accumulate, uptake cadmium from a solution. And although it will mainly stay in the roots, the plants are able to translocate it to the flowers and to leaves and stems as well. So photochemical efficiency for this, we used a mini PEM. It's what the equipment is called. And it just measures the ratio between the photosynthetic efficiency when all the centers are closed versus when all the centers are open. And there's more detail on the methodology in the published paper if you want that. But what we could see is that for 0, 2.5, and 10 milligrams per liter of cadmium treatments, there was no difference, no reduction in photochemical efficiency. But then for the 25 milligrams per liter of cadmium, there was reduction in photochemical efficiency. And this was conducted at 2 time points, so 14 days after treatment and 45 days after treatment. This 45 days after treatment, it's when this treatment, the 25 milligrams per liter treatment, was harvested. So it makes sense. In the picture, you can see that the plants were chlorotic and necrotic, so it would make sense that the photochemical efficiency in those plants was lower. We also measured chlorophyll content index at 2 time points, the same time point. So on the A and B are 14 days after treatment, and C and D are 45 days after treatment. And the one circled in green is for experiment 1, while the one circled next is going to be for experiment 2. So you can see that in the beginning, at 14 days after treatment in figure A, 0 and 2.5 milligrams per liter cadmium showed no significant differences, and then it got a little lower for 10 and lower for 25. But then 45 days after treatment, it seems like the 10 milligrams per liter cadmium kind of bounced back, because there was no significant differences between 0, 2.5, and 10. Only the 25 treatment was lower. And then for experiment 2, we can see the same thing happening, although at 45 days after treatment, the 10 milligrams per liter of cadmium treatment was still significantly lower than the control. So total THC and CBD concentrations, we also have experiment 1 and 2 in this table. And we sent out our samples for cannabinoid analysis. It's a lab in Macon. And you can read more about the methodology in the paper as well. But we saw no differences in experiment 1 for either CBD or THC among all the treatments. In this case, only the 3 treatments, because as I said, the highest cadmium treatment did not produce any flowers. But then in experiment 2, we saw a decrease. We saw a decrease in CBD and THC in the 10 milligrams of cadmium per liter. So it was inconsistent between experiments. So now the transcript abundance of metal transporter and cannabinoid biosynthetic genes. So we wanted to identify heavy metal transporters in cannabis sativa. So for that, we retrieved ortholog sequences from Arabidopsis from this P1B type ATPase gene family, and it contained 8 genes, which we decided on those genes based on previous research on this topic. And then we analyzed spatial and temporal expression patterns of the genes coding for metal transporters, which in cannabis sativa were HMA1, HMA3, HMA3C, which is the complement, HMA4, HMA5, PAA1, and PAA2. And we analyzed those genes in roots, leaves, and flowers of the plants exposed to all the cadmium treatments at 3 time points, 2, 45, and 68 days after treatment. Here's a list, and in the paper, there's all the primary sequences as well. You can look at that in the paper. We also analyzed the cannabinoid synthase genes, which are the THCAS and CBDAS, and we used 2 reference genes to normalize the data. So here's a heat map. So basically the way to look at a heat map is the more red, it means higher expression. So here we have leaf versus root for all the treatments. And this is at 2 days after treatment. So you can see this is just for you to have a better way to visualize that all the metal transporter genes analyzed were generally higher expressed in roots compared to the leaves. So this is just a cool way to see it. I love heat maps. And here's another one. So this one, it's only for root tissues, focusing on the roots, since most of those transporters were highly expressed in roots. So we compared all of them throughout different time points. So we have 2, 45, and 68 days after treatment. And we can see that the ones that were highly expressed throughout all the experiment were the HMA5 and PAA2. And here is just a figure from the published paper, and I'm gonna just make it bigger, but just so you have an idea, we analyzed the relative expression for all the genes and in roots. This is all for roots, and in all the time points. So 2, 45, and 68 days after treatment. And what we found was that HMA1 and HMA3, as you can see, at 68 days after treatment, so this was at harvest, the expression of these genes, these metal transporter genes, in plants under the 10 milligrams of cadmium per liter, they were upregulated at harvest, HMA1. For HMA3, they were upregulated at harvest and at 45 days after treatment. For HMA4, the same happened at harvest. 10 milligrams per liter caused this upregulation in this transporter gene. And for HMA5, the same happened. So you can see how, different genes will be differently expressed throughout the experiment. For THCA and CBDAS, which are the cannabinoid synthase genes, first, we didn't see anything happening related to CBDAS expression at any time point, but then THCAS, we saw that the treatment of 10 milligrams of cadmium per liter caused a decrease, in transcript abundance of THCAS at harvest. So a few conclusions. We saw that cannabis sativa can tolerate the exposure of 2.5 and 10 milligrams per liter in a hydroponic solution. We saw that the plants didn't show any decrease in plant height or biomass, in dry weight. And in one of these experiments, we saw a decrease in CBD and THC. However, in the first experiment, we didn't see anything. So this is inconclusive. Nevertheless, the 2.5 milligram per liter of cadmium, it showed no differences in CBD or THC. Concentrations of 25 milligrams per liter cadmium severely restricted growth, reduced photochemical yield, chlorophyll content index, and caused premature death. Didn't even produce flowers or anything like that. The cadmium concentrations were highest in roots, but there was significant accumulation of cadmium in leaves, stems, and flowers. And the concentration in plant tissues increased with increasing cadmium in the solution. Plant biomass and total CBD and THC in flowers were not affected, in the controlled treatment, and plants exposed to up to 10 milligrams per liter of cadmium, like I mentioned before. And the transcript abundance of HMA1, HMA3, HMA4, and HMA5 in the roots were upregulated in the 10 milligrams per liter of cadmium treatments, suggesting that this metal transporter genes may be associated with cadmium uptake, sequestration in roots, and xylem loading. And while the concentrations were used in this experiment were a lot greater than what would be encountered in egg soils, as I mentioned before, our results just show that the hemp plants have the potential to accumulate cadmium in the flower tissue, which was, our concern, especially with hemp being used medicinally. So these are some of our conclusions. It just emphasizes the need and the importance for heavy metal testing in hemp. And I think that the heavy metal testing should be conducted throughout production, throughout any type of media they use, any type of solution, if it's hydroponic experiment, if it's in the field, if it's in greenhouse. It's important to test your soil, test your media as well, and then test your final product, that being biomass or being smokable flower. It's very important to pay attention to this kind of stuff. And yeah, I highly recommend everybody to read the paper. If you have any questions about the methodology, and the results are much more detailed in the paper, of course. I didn't spend a huge amount of time going through the details, but you're welcome to take a look at the paper, and you can reach out to me if you have any questions. And yeah, I would love to reply to any questions you have. Just shoot me an email. I'll leave my email here at the end of this presentation, aom@uga.edu. And yes, I'll be graduating this summer. And like I said before, I'm very thankful for Dr. Justice for, helping us with the plant genetics and helping us with a lot of questions we had throughout the experiments. And I really appreciate the chance of talking a little bit to you guys, presenting my research, and I'm excited to know what you think about it. So thank you so much, and talk to you soon.